Uh, Thank you again. We now move on to this lecture referred as gluconeogenesis. It is basically how to attain glucose within your body without relying on diet glucose. Also that of storage glucose from glycogen. It's making glucose within your body itself by using 2 carbon, 3 carbon intermediates that become precursor for this gluconeogenesis pathway. So before we go into the gluconeogenesis pathway, we need to understand the meaning of anabolic pathway as well as catabolic pathway. In terms of gluconeogenesis, it is under the anabolic pathway that is making large molecule, that is the 6 carbon glucose, from using 2 carbon lactic acid, 3 carbon pyruvate, and many others. So it is from small molecule to larger molecule, and when that happens you need to provide energy because bonds are being made from say 2 carbon to 3 carbon, eventually become 6 carbon, and these bonds need energy, and the energy is actually supplied by NADH and more likely of NADPH as well as ATP. And then you also have catabolic pathway being defined here, that is large molecule to smaller molecule. And this one example is glycolytic pathway, where the 6 carbon sugar glucose being oxidized completely to become carbon dioxide and water. So glucose 6 carbon become 1 carbon dioxide. That means there is a loss of energy found in the 6 carbon glucose, and this energy is attained in the making of ATP, reduced coenzyme of NADH, reduced coenzyme of FADH2, as well as release as heat as well. This slide basically looks into overview of this pathway itself, which I will again repeat later in detail. But basically, gluconeogenesis pathway is to synthesize glucose, and it happens mostly in liver, 80% in the liver, and the rest in kidney. And as I referred to you earlier, it requires 2 carbon and 3 carbon, and these are the intermediates shown here: lactate, pyruvate, glycerol, and amino acid. In fact, there are others as well, and there are key enzymes required to make bypasses to reverse the process of glycolysis, to be said literally. And then, what is the purpose of gluconeogenesis? That is to make glucose in our body is during fasting and starvation conditions in general. So you have here the definition of gluconeogenesis. It is to make glucose from pyruvate as well as the other precursors mentioned in the slide before this. And in terms of the word itself, gluconeogenesis, it is to neo means to it's sugar, and then genesis means create. So it is to create sugar in general terminology itself. And on the left here you have glucose to pyruvate, and as well as pyruvate to lactate. Um, when it goes from glucose to pyruvate and then later to lactate by anaerobic glycolysis, so you can say that this gluconeogenesis is actually a direct reverse of glycolysis. But however, you have to pay attention that there is compartmentalization, where the process of gluconeogenesis happen in the liver as well as kidney, while glycolysis happen in the myocyte cells and also other cells, as well as even in liver and kidney. So if it happens, glycolysis and gluconeogenesis together in the liver cells, then you have an issue where something referred as futile cycle could take place, and that's not energy efficient altogether. So here you have the main path, that is from pyruvate after a number of step finally become glucose. So it's reverse of glycolytic pathway, the 10th step of glycolytic pathway, but because of certain steps in glycolytic pathway which is considered irreversible, therefore you need to have new sets of reaction which need to bypass these irreversible steps itself. And also here shown where all the other intermediates such as lactate, amino acids, propionate, glycerol, even the other monosaccharide fructose and galactose come into this main path in order to finally achieve glucose itself. So here we cover the steps in briefly first, and then later we go in much detail in another slide. It starts off with lactate, but not necessarily start off with lactate. Also have other precursors coming in order to be the starting material for this gluconeogenesis. So this is, if you look at it, you will realize it's actually glycolytic steps itself, and it's going in reverse. Okay. Then you have these three red arrows, which are irreversible steps of glycolytic pathway, which that the meaning of irreversible means you cannot go back from pyruvate to phosphoenol pyruvate, or fructose 1,6 bisphosphate to fructose 6 phosphate, or glucose 6 phosphate to glucose. So in order to go back, you need to bypass these steps, these crucial steps, which are actually the key regulatory enzymes found for glycolytic pathway, that is hexokinase, phosphofructokinase, as well as pyruvate kinase. So it starts with lactate and then goes to become pyruvate, and then bypass is done to become oxaloacetate first by one enzyme, and then followed by another enzyme or another step converting oxaloacetate to phosphoenol pyruvate. We also have another bypass. This one only need one step rather than the two steps shown for pyruvate to phosphoenol pyruvate. And then you also have one more step to bypass from glucose 6 phosphate to glucose. And then the enzymes relevant to bypass are known as phosphatase, because they remove phosphate. You also need to understand that three kinases are the one that is relevant for the irreversible steps, that is exokinase, phosphofructokinase, and also pyruvate kinase. And these are the one that need to be bypassed. However, even though there's another kinase, but this kinase is a reversible step and no need to be bypassed, because it can just go from 3-phosphoglycerate glycerol acid to 1,3-bisphosphate phosphoglycerate acid. These are the irreversible glycolytic steps that I was referring to. The three key regulatory enzymes, that is hexokinase, phosphofructokinase, as well as pyruvate kinase. And these enzymes are irreversible, therefore must be bypassed rather than just reverse the reaction altogether. And to be able to do that, you need enzymes such as glucose 6-phosphatase to reverse the or to bypass the exokinase step. And then you need fructose 1,6-bisphosphate to bypass the phosphofructokinase 1 step, followed by two enzymes, that is pyruvate carboxylase followed by phosphoenol pyruvate carboxykinase to reverse the step catalyzed by pyruvate kinase itself. This is gluconeogenesis, also defined as the synthesis of glucose de novo. That means from scratch. That means de novo means you are making 2 carbon, 3 carbon, putting them together and eventually become the 6 carbon glucose. It's not like, you get from the diet 6 carbon glucose and then you get glucose altogether. But this one is you build it within the cell, within your body itself. That's why it's referred as de novo. And then it is an anabolic pathway for the synthesis of glucose from many small precursors, not only lactate. In terms of its importance, it is primarily in liver, 80%, and kidney. So you have to understand, it is also in the liver you have glycolytic pathway. However, it is being regulated in order to have only one pathway happening at a time. But in the muscle cell, for instance, there is no gluconeogenesis. Therefore, it is compartmentalized, so it's safer that way. And then its important is primarily in ensuring blood glucose level is kept at a certain concentration in your blood. If the blood glucose falls too low, it is possible a person could end up fainting because there's not enough glucose for the brain itself. And if the glucose is too high, and cannot be controlled, and then the person will have an issue where they might have disease referred as diabetic, for instance, because too high of a blood glucose cannot be controlled by secreting insulin. So when the issue is when the blood glucose falls to a dangerous level, then gluconeogenesis happening in the liver able to produce glucose, free glucose, and this free glucose can be put into the bloodstream to elevate the blood glucose back to normal state altogether. And that's the purpose of this gluconeogenesis during times when the blood glucose level drops. This Cori cycle is basically a cycle that takes lactic acid from, for instance, muscle cells that are undergoing anaerobic glycolytic pathway, and then they release lactic acid, and that lactic acid cannot be converted to pyruvate and used by muscle cell itself. Therefore need to be secreted into the bloodstream, and the Cori cycle begins by taking this lactic acid, all the lactic acid from myocyte, red blood cell to the liver, and then by gluconeogenesis the lactic acid is converted to become glucose, and then the glucose is dumped into the bloodstream and it is brought back to the muscle cell. So that's basically Cori cycle, and this gluconeogenesis is plays a main role for the Cori cycle itself. So here we go through the gluconeogenesis pathway in much more detailed understanding. So it's got two stages, stage 1 as well as stage 2. We begin by having lactate becoming pyruvate, and then become oxaloacetate. Right? And then, in actual sense, you can categorize gluconeogenesis having been done in cytoplasma. But this one step, because oxaloacetate is intermediate found in the mitochondria, therefore pyruvate will be go into the mitochondria, become oxaloacetate, and then it needs to come out. Oxaloacetate cannot come out easily from mitochondria. Therefore, it is made into malate, and through malate aspartate shuttle, it can then come out. And then another enzyme react on it, that is PEP carboxy kinase, to convert oxaloacetate to phosphoenol pyruvate. So we can still generalize that gluconeogenesis happen in the cytoplasma overall, rather than taking into account this single step done in the mitochondria. Next thing, you also have other precursors can also come in to this gluconeogenesis pathway, such as L-alanine, glycerol. Glycerol is intermediate that comes about after breakdown of triacylglyceride. You know, the backbone of triacylglyceride is glycerol. So glycerol also can come in into the dihydroxyacetone phosphate and then continue to make glucose. You also have L-alanine as well as L-aspartate. These are carrier amino acids, where the 20 amino acid can also contribute. Certain 20 amino acid can contribute towards making glucose in human. Therefore, they also come in these areas to be part of gluconeogenesis, that is stage 1. We move on to stage 2, where the bypass takes place at this location here for phosphofructokinase 1. This one, nothing to describe any further. It's quite straightforward. Here you have to understand that pathways are not standing alone. In fact, they are connected to other pathways in our body. And here is an example where once the kidney or liver generates a lot of glucose 6-phosphate by using all these 2-carbon, 3-carbon intermediates, there will be excess of glucose 6-phosphate, or even excess of glucose. What to do with it? So if there is excess of glucose 6-phosphate, it can be made into glycogen by entering the glycogen metabolism itself. And then when there is a drop in glucose in the liver, and also liver requires it to dump glucose into the bloodstream, the storage glycogen can also be converted back to glucose 6-phosphate, and then followed by this bypass by another phosphatase enzyme, that is glucose 6-phosphatase, and then you get free glucose, and this free glucose can then supply glucose to the bloodstream, or even glucose for glycolytic pathway of the what do you call the liver itself for energy. So here, just to highlight the fact, there is 3 irreversible reaction, and one of it is having a very high, 3 times more Gibbs free energy compared to the other 2. And this is an indication why it needs 2 different steps, 2 different enzymes to bypass this pyruvate kinase, because it has the flux forward, or the conversion of PEP to pyruvate requires so much of energy release. So in order to bypass, you need two reaction steps. And just to highlight that fact altogether by using these values. This one is about mechanism of this enzyme, and to stick with my principle that I'm not going to assess you on the mechanism of enzyme. I'm not going to cover this at all. I'm going to quickly move forward to the next slide, which is much more relevant than this few slides here. This is basically a contribution of the coenzyme that is found, the biotin for the enzyme, and with a swinging arm that helps to take from one active site to the next active site. So the enzyme has more than one active site to complete its reaction where it converts pyruvate to become oxaloacetate. So once the oxaloacetate has been made by the effect of that enzyme just now, it will then be converted to phosphoenol pyruvate, and surprisingly it requires energy, but not in the form of ATP but using GTP. The hydrolysis of GTP helps in the conversion of oxaloacetate to phosphoenol pyruvate, and also it requires a decarboxylation. That means the number of carbon in oxaloacetate will be reduced in order to become 3-carbon PEP. The rest is actually covering the other two steps, that is the other two bypass steps. So now we answer this question that why do we produce glucose in our body? Is there a need that we need to come up with glucose that need to be made from scratch, de novo, rather than relying on diet itself, or rather than relying on glycogen that is stored in our body? The reason behind the body makes glucose itself is simply because of the brain and also the blood or red blood cell that requires the plenty of this glucose. So whenever the body or blood glucose level drops into an alarming concentration, it will be a huge problem, especially the brain. If the brain or the blood sugar drops somewhat below 2.5 millimole of concentration in the blood, it could lead to a devastating effect where the brain do not have glucose, and therefore it could end up causing the person to be fainting and become coma and even eventual death if it's not the glucose is not restored. And also red blood cell rely mostly on glucose because it doesn't have mitochondria. So it needs to be constantly doing glycolytic pathway in the manner of anaerobic glycolytic pathway. And therefore, these two components of your body must have always sufficient amount of glucose. And for that purpose, the gluconeogenesis, also glycogenolysis happening in the liver helps to ensure there's always good glucose level in the blood itself, normal glucose level. In fact, these two components of the body is been designed so that they don't rely on insulin. They are not affected by insulin, and therefore glucose, whenever glucose is present around them, glucose can be taken up without the need of insulin binding to the insulin receptor altogether, because they are crucial component of your body. So now that we already established the answer for that question, we look into the importance of the liver. So liver is where all the gluconeogenesis takes place, and it's actually fed with not only pyruvate, also lactic acid that comes from red blood cell, as well as skeletal muscle that is undergoing anaerobic glycolysis. So this is covering all the 2-carbon and 3-carbon precursors that become the starting material for gluconeogenesis. You have lactate coming from muscle, coming from red blood cell, and then it goes into gluconeogenesis in the liver and become glucose. You also have certain amino acids that will be, you have 20 amino acids. They can be either free amino acid or also found in tissues or in muscle that can be broken down in order to get the free amino acid, and it can be then brought as adenine, and in the liver it can also be playing a role to make into glucose. So you have a situation where from protein you can get carbohydrate. You also have citric acid cycle intermediates or Krebs cycle intermediates, where you have oxaloacetate, you have alpha ketoglutarate, where the 20 amino acid, some of them can be undergoing transamination or transaminase reaction in order to take away the amino group and then convert to oxaloacetate as well as alpha ketoglutarate, and they can also rush into gluconeogenesis in order to make glucose. This propionate is one of the product of odd chain fatty acid oxidation through beta oxidation, where it will cut 2 at a time, and then you have leftover of 3 carbon, which will be made into propionic acid, and that can enter into the citric acid cycle as succinate, and then become what is referred as glucose by gluconeogenesis. Then you have also glycerol, you know, the triglyceride. You take away 3 fatty acid, you have glycerol that also can be made into glucose. This again, just to repeat back what has been said before. You have lactate. You cannot convert a lactate back to pyruvate and then enters citric acid cycle in the muscle cell. It cannot be done. So lactate has to come out into the bloodstream, go to the liver, and then it can be further oxidized. It can be made into glucose and then brought back to muscle and then further oxidized in the muscle. So that is the only way. You cannot use the lactic acid. So here we take a particular activity of our body, that is muscular activity, to understand the contribution of gluconeogenesis. So muscle contraction requires ATP hydrolysis, that is requires energy. Where do we get the glucose for the muscle activity? In order to get ATP, that is coming from free glucose, as well as the storage glycogen, where glycogen undergoes the pathway of glycogenolysis. you will get free glucose. And then what happened to the glucose? It's either go through aerobic if there is plenty of oxygen, but if there's not much oxygen, demand for ATP is very high in the contracting muscle, then it will go through anaerobic, and then you have buildup of lactic acid. The lactic acid fate is already go back to liver and then converted to become glucose. Here, referring again about the lactic acid that is made in the muscle cell, being brought to the liver by this Cori cycle, and then lactate become pyruvate and then become glucose with several steps in the gluconeogenesis pathway. And then this is when the muscle really requires the glucose. When the muscle start to relax, requires no more activity, that means no need contraction, what happens? Now you have a situation where excess of glucose is present, and this glucose will be immediately converted to glycogen and stored in the muscle cell itself. Even any newer glucose coming from liver will be just made into glycogen and stored in the muscle cells. The last sentence actually referring to the importance of Cori cycle, where if more and more lactic acid build up in your muscle cells, it will cause acidosis, a condition because of high acid become very acidic and therefore can be very bad for the cell. So this Cori cycle is very, very important to remove this acid condition by taking the lactic acid to the liver itself. Here we give the detailed description about the Cori cycle itself, where it starts off, or it can be starting either from the liver or from the muscle. Here give an example from the liver. So you have plenty of lactic acid coming from muscle, red blood cell. You need to convert it through gluconeogenesis to become glucose, and then this free glucose can be dumped into the bloodstream and then be brought to the muscle cells. When in the muscle cell, glucose is utilized through glycolytic pathway, and at certain times will be made into lactic acid by anaerobic glycolysis, and then if it stays there, it will become acidosis. Therefore it is then brought out through the bloodstream all the way to the liver and then made into glucose by gluconeogenesis. So this is Cori cycle, simply removal of lactic acid from muscle to liver. Here we see the significance of gluconeogenesis under two conditions, that is starvation and fasting. Starvation is when you are, you are forced to not take any food, and you have no control on when you can start eating again. While fasting is something of a religious, mostly religious purposes, and also can be also due to medical condition. If you need to do some diagnostic tests, you also go through fasting. Fasting is much more control, where you can at any time choose not to continue with fasting. But starvation sometimes is beyond your control, and it can be prolonged even up to death itself. So under these two conditions, this is where the pyruvate oxaloacetate can be changed into glucose, and the source of this pyruvate and oxaloacetate could be from amino acid breakdown, that is after removing the amino group. And where do you get this amino acid? It is either found free in your body, in your cell, or also connected to tissues like muscles. It is only very bad situation, usually during time of starvation, where no choice, no more of fatty acid, no more of carbohydrate, where the body finally resort in taking the muscle proteins and then consider muscle wasting in obtaining amino acid and using it to form glucose to stay alive, at least in the final stages of starvation. And then you also have glycerol that can be utilized, that comes from the adipocyte, where lots of triglyceride is present, where you can remove the fatty acid and use it through beta oxidation to get energy for the body during time of starvation and fasting. But also possible to use the glycerol, but not as effective as the fatty acid, where glycerol can be changed into glucose to support during time of starvation and fasting itself. So due to time constraint, and also I'm not going to touch much on the regulation part in exam, so I'm going to go fast here, just get to the point. Basically, you have synthesis, that is making glucose, gluconeogenesis pathway. Then you have glucose to pyruvate, the glycolytic pathway, degradation. These two cannot happen together. It is possible to happen in the liver together because both of these pathway are present there. But in the muscle, no worries, because muscle, there's no gluconeogenesis. It's basically compartmentalized. In the liver, if it happens together, then you have a big issue where you will have to give more energy in order for uh, synthesis to take place. That's why you need to have these two, synthesis and degradation, to be happening reciprocally. One should be on and the other one should be off. This final slide actually tells you about what happens when both of the pathway happens together at the same time. That is spontaneously. If it happens at the same time, it is referred as being futile. Futile cycle, that means it's not beneficial for the system itself. If glycolysis happens, you will get 2 ATP, or high energy phosphate. If gluconeogenesis also happen together, then you will get 6 high energy phosphate, that is 4 ATP plus with 2 GTP, equivalent to 6 high energy phosphate. But if both happen together, you have a situation where 6 minus 2, you need to provide another 4 ATP every time these two pathways takes effect or happens spontaneously. That's why it's not possible, because more energy needed to be delivered to the system rather than you can get ATP from the system. That's why it is called futile cycle. In the body, it's possible that more of glycolysis as well as other catabolic pathway happens before any anabolic pathway can go through.